Good afternoon. My name is Kat Bryant-Knudsen, and I'm the founder and president of the Speak Foundation. Thank you to the LGMD community for tuning in today. The Speak Foundation is the first patient-led organization for limb-girdle muscular dystrophy, and our mission is to speak up for those without a voice and to provide hope to those living with muscular dystrophy. For those who don't know, we lead a variety of initiatives, including the LGMD International Conference, the LGMD News Magazine, and the LGMD Patient Network, where you can add your name and information to receive community updates. We are thrilled to welcome Sarepta Therapeutics and host their webinar on Imagene, Sarepta's investigational gene therapy for LGMD 2E/R4. This community webinar is live today, and we encourage your questions during the live Q&A portion. We will first start with 30 minutes of presentation from Jessie Coy, Vice President and Global LGMD Program Team Leader. And with that, I will turn it over to Jessie.
Thanks, Catherine. Hi, everyone, and thank you to the Speak Foundation for hosting today's webinar. And thank you to everybody for joining today's conversation. As Catherine said, I am Jessie Coy, and I lead a multidisciplinary team at Sarepta, where we are focusing on gene therapies for limb-girdle muscular dystrophy. For today's webinar, I will be presenting you with information and overview of Sarepta's work in limb-girdle muscular dystrophy, with a specific status update on SRP-9003, our program for limb-girdle type 2E. This presentation contains forward-looking statements. These forward-looking statements involve risks and uncertainties, many of which are beyond our control and are based on our current beliefs, expectations, and assumptions regarding our business. Please do take a moment to review our slide, which contains our forward-looking statements. Within our pipeline, Sarepta is working on 6 of the limb-girdle subtypes. The treatments we are developing are gene therapies that are unique to each of the subtypes for which no current approved therapies exist. We anticipate if we are successful in delivering these programs, then we will be addressing approximately 70% of the current known limb-girdle muscular dystrophies. All of the programs are in various stages of development, which I will cover later in the presentation. Before we go into our programs, let me spend a little time talking about limb-girdle muscular dystrophy and why knowing your subtype is particularly important. The limb-girdle muscular dystrophies are a group of inherited neuromuscular diseases. They are referred to as monogenic conditions as they are affected by a single gene. It is a condition that affects both males and females and is considered collectively as a rare disease affecting an estimated 1.63 per 100,000 persons. The LGMDs are caused by a broad range of mutations in the genes that encode proteins that control muscle function, regulation, and repair. The muscles affected by limb-girdle are those primarily in the shoulder and pelvic area, so those large muscles on the top of your arms and the hips and the thighs. The limb-girdles are caused by a broad range of mutations in the genes that are important to ensure muscle function. These genes that are mutated affect the instructions for making the proteins. These proteins are critical to support processes like how a muscle performs its function in the body. If we look at the top graphic, when a gene is not mutated, there is clear instructions on how to make the proteins needed. Now, looking at the graphic below, if there is any change in that gene, what we end up with is either a protein that is made but cannot perform its function or is not produced at all. So what parts of the muscles are we referring to? Our muscle membrane is called the sarcolemma. The role of the sarcolemma is to protect the muscle fibers. As shown here, like electrical wires are protected by insulation, such is the role of the sarcolemma for the muscle fibers. Looking at the skeletal muscles on the right, you can see that the sarcolemma is protecting each and every muscle fiber in the entire muscle. Focusing now on the muscle fiber, we are demonstrating here the impact of a loss of beta-sarcoglycan, which is a protein found in the sarcolemma, and how this loss affects muscle function. Without the beta-sarcoglycan, the dystrophin-associated complex is affected, and without a way of resolving this, then the sarcolemma continues to be damaged over time, leading to eventual muscle destruction during the progression of the LGMD. Now, not all the affected proteins for the LGMD are on the sarcolemma. They can also be from inside the muscle fiber too, which can also ultimately impact muscle functionality. We have outlined here the different LGMD subtypes and which protein in the muscle it relates to. At Sarepta, we are working on investigational therapies that deliver a DNA construct that expresses missing or non-functional proteins. The construct consists of the vector, the promoter, and the transgene. The virus vector that was selected for our construct is AAV-RH74 and is based on our experience with this vector's safety profile and its efficiency in reaching the cells. The next piece of the construct is the promoter, and this ensures that we reach the right cells needing this protein. The final piece of the construct is the transgene that aims to provide cells with the instructions for making the therapeutic protein. You may also want to refer to the gene therapy FAQ information featured in the new LGMD news magazine, and our Sarepta team would be more than happy to share resources with anyone liking to learn more about gene therapy. Now, before infusion of the product, it is prepared under special conditions at the hospital and made ready for intravenous infusion. As the product enters the cells in the body, it begins to do the work of creating the intended protein. It is important to note that due to the construct of the gene therapy, it is a one-time only therapy. As depicted earlier, if a gene is missing in LGMD, it will affect how the muscle functions. The goal of gene therapy is to stabilize or slow down the disease. So when you get the gene therapy, you may already have the disease affecting your muscles. And the aim is that once the gene therapy is introduced, you will have a slower progression or stabilization of your LGMD. So in the case of beta-sarcoglycanopathy, or LGMD type 2E, we are providing the beta-sarcoglycan gene with Sarepta's SRP-9003 construct to restore the dystrophin-associated protein complex and ensure that what was missing is now restored. There are many checks that are carried out to ensure if you are eligible for a gene therapy trial. One of the requirements is that we have to ensure that there is confirmation of the specific LGMD subtype through genetic testing. Genetic testing is far less invasive than it used to be and can be done using blood or even saliva samples. Please talk to your doctor or genetic counselor to get more information. Genetic testing technology has advanced in recent years, and there are many tests and programs available in the US that offer genetic testing for eligible individuals. Again, please speak to your healthcare provider to learn more if you have an interest in a genetic test to determine your subtype. I'm now going to spend some time going over Sarepta's investigational gene therapy, SRP-9003. SRP-9003 is a product still in the research stages of development and is not yet approved by any regulatory authority. It is being developed to treat patients that have been diagnosed with beta-sarcoglycanopathy, but it's also referred to as LGMD type 2E or R4. Let me first cover the drug development pathway before we go into the specifics for SRP-9003. All drugs are initially developed in the laboratory. During this stage, there are many tests that are performed to ascertain whether the drug will work as we anticipate, meaning, is it working through the pathways we want? This stage is called proof of concept. There are many different iterations of a product that is tested during this stage before we can confirm that it is suitable to test in animal models. Here, the product undergoes various different tests in these animal models to assess whether it is safe and not causing any harm to major organs nor unintended physiological effects. In addition, we have to verify that it is having the desired effect of what we want it to do in the body. These tests can take many years to perform because we have to test and monitor the product in these models over time to prove that it is working like we intend and that there is no safety concerns. We then have to decide what would be the most safe and efficacious dose to take into the clinic. Only once we have completed all of those tests and provided assurances to the regulatory authorities that the product is suitable to be tested in humans are we allowed to initiate clinical trials. Often, time to reach this stage can vary from 5 to 15 years, depending on what we learn through research and the preclinical stages. With gene therapy, we have to provide a high degree of certainty that the product is suitable for patients, since this is a single, one-time only treatment that the patient receives. During early clinical research, we provide assurances to the regulators that the drug is safe before we start late-stage studies. It is during late-stage clinical research that the sponsor has to demonstrate with a high degree of certainty that the product is both safe and effective for use. Now, in parallel to all of the clinical work, extensive research and development activities are being conducted on manufacturing the product and ensuring we have the most appropriate tests to assure product quality before it is released for use in humans. It is with this preclinical, clinical information, together with those extensive details on how the product is made and tested, that a sponsor can then submit to the regulators for requesting registration. Only after approval can a product be then placed in the market. Work still continues even beyond that, where post-marketing studies occur to continue to provide regulators with further information on the product. Sponsors like Sarepta keep the patient's safety at the forefront of all our activities to ensure we take a very considerate approach through each of these development stages for each and every product that we develop. Now, focusing on the development of SRP-9003. This program has passed the preclinical stage and we have completed early clinical studies. We have 2 studies that we have performed in early research. The first study, which we call Study 101, was a study where we evaluated the safety of SRP-9003 in young ambulatory patients. We then conducted another study, 102, called Imagene, where we are evaluating the safety, tolerability, and efficacy of SRP-9003 in both ambulatory and non-ambulatory patients. In addition, because LGMD is such a rare condition, we have to develop a better understanding of how the disease progresses, and therefore, in addition to the clinical studies, we have conducted a global natural history study in LGMD type 2E. This study does not involve any treatments, and we follow the patients over a period of time to understand how their limb-girdle progresses. Today, we are going to focus on our late-stage clinical study, 301, which is Sarepta's phase 3 multinational open-label systemic gene delivery study to evaluate the safety and efficacy of SRP-9003 in subjects with limb-girdle muscular dystrophy type 2E or R4. This study has been named Imagene. In Imagene, individuals that enroll in the study will participate for up to a period of 5.5 years. Study visits are frequent at the beginning of the study and decrease over the duration of the study. Subjects enrolled in the study will have further details provided to them by their physician. Additionally, eligible individuals will have details of study assessments that are required to be performed in the study. These include, but are not limited to, physical examinations, laboratory tests, functional assessments, muscle biopsy, MRI, and the use of wearable devices. Once successfully enrolled in the study, you may need to undergo a lead-in period, where we obtain some initial pre-treatment data on how your disease is progressing. If, however, you have already participated in the journey or GRASP studies, we will use that data. and you can go straight into the treatment stage of the study, where all eligible participants enrolled in the study will receive SRP-9003 drug product. Imagene is an open-label study, which means that all patients receive the study drug and there is no placebo treatment in this study. In Imagene, we plan to enroll approximately 15 participants. There are two groups or cohorts in the study. There is an ambulatory group and a non-ambulatory group. It is open to both males and females that are greater than or equal to 4 years of age, and there is no upper age limit in the study. However, we do have eligibility criteria that will need to be satisfied in order to participate in the study, and I will go into that into more details in the next slide. In order to participate in Imagene, participants have to satisfy the criteria outlined here. You have to ensure you meet the inclusion criteria and also demonstrate that none of the exclusion criteria applies to you. As I mentioned, we have two groups in the study. So to be included in the ambulatory cohort, you need to be able to walk without assistive aid, perform the 10-meter walk test in less than 30 seconds, and have a score of greater than or equal to 25 in the NSAD. The North Star assessment for limb-girdle type muscular dystrophies, or NSAD, is a metric that involves performing tests that can be related to daily activities for ambulatory patients. In the non-ambulatory group, participants perform a different group of tests as outlined, and this includes a test called the Performance of Upper Limb, or PUL 2.0, which is a test relevant to the daily activities for non-ambulatory individuals. Your physician will provide further details on these tests should you decide to consider participation in the study. All patients in either group must provide evidence of beta-sarcoglycan genetic mutation, be able to cooperate with the required muscle testing, and since this is a gene therapy, individuals have to demonstrate that they do not have elevated antibody levels to AAV RH74 vector. In terms of exclusion criteria, participants must have their heart and lung functions assessed and ensure that they have no other significant conditions. This eligibility criteria helps minimize interpatient variability, which can sometimes impact the study data. You may want more details than we can cover today, and your study physician will be able to provide details of all the inclusion and exclusion criteria and what the screening process will involve should you consider participating in the study. In order to demonstrate that the SRP-9003 investigational drug is performing as intended, we aim to demonstrate that it is safe for all patients that receive the drug in the study. For this, we will monitor participants throughout the study for any adverse event that they may experience throughout the study. In addition, we aim to demonstrate that the product is efficacious by showing that following infusion with SRP-9003, that the body produces beta-sarcoglycan protein in your muscles. That is why we have to do the muscle biopsies. Finally, we aim to demonstrate that after treatment with SRP-9003, there's an impact on those functional assessments that I outlined earlier. That is why we have to do the functional assessments. Now, for other practical considerations in Imagene, we acknowledge that participating in a study requires time and commitment, and our goal is to minimize the burden. Therefore, as part of the study, we have arrangements in place that for study visits, participants will receive a reasonable travel and meal reimbursement. A study travel vendor is available to support with travel arrangements to the study site. And dependent on local regulations, there may also be an eligibility for a financial stipend. Sarepta cannot support relocation of any kind, nor visa applications to participate in this study. It is important that individuals considering participating in a clinical study take careful consideration when assessing their options to participate. Please discuss this with your family and physician as you make your decision. We have outlined here some trial-specific considerations for Imagene. SRP-9003 is investigational, and the safety and efficacy is not yet established. Gene therapy is currently considered a one-time therapy, and there is currently no redosing option. There is long-term data that is needed in the study, and therefore participation over several years with 5 years follow-up post-dosing. Participating in the study could limit future treatment options and participation in future clinical trials. There is a significant time commitment that will be required, and this may be considered burdensome for participants, the caregiver, and the family. Please take all of this into consideration when you discuss your options with your family, your physician, and your care team, if you are considering participating in the clinical study. For Imagene, we have outlined some of the key steps in the study. After you have considered your options and you want to consider participation, please reach out to your doctor. They will advise on how to find an appropriate site that is conducting the study. Individuals will then have the opportunity to speak to the study site coordinator to understand more about the study and the eligibility criteria to participate. A screening appointment will be scheduled, and this is when inclusion and exclusion criteria is discussed. If an individual has consented to enroll in the study and has satisfied the eligibility criteria, then they can enroll into Imagene, and then further tests are performed, which includes the muscle biopsy. Once all of these steps are completed, including a 6-month lead-in, then participants are dosed with SRP-9003. After dosing, there is a series of follow-up visits that vary in frequency. There are then post-treatment biopsies that are also performed. Details of these and all other follow-up tests will be provided to participants during their enrollment discussions. This study has a 5-year follow-up post-dosing, so please expect study visits throughout this period. This follow-up period is a very important step in the development and data gathering process during the drug development to inform on the treatment expectations and to understand the safety profile of SRP-9003. To find out more, clinicaltrials.gov is a database of global studies. It is a requirement that details of a study like Imagene is posted on a site like clinicaltrials.gov. This database is a resource for finding clinical trials. You have the option to use various criteria, including the name of the disease, a particular study drug, or a study identifier. We have provided the study identifier for Imagene on this slide, together with a reference QR code. You can use clinicaltrials.gov to gain up-to-date information on Imagene. As I previously mentioned, Sarepta has 6 programs in various stages of development for LGMD. All these products are in development and none are currently approved by any regulatory authority. Work continues across the portfolio, and we aim to bring more therapies into the clinic in the near future. I briefly mentioned our natural history study earlier as part of our development plan for SRP-9003. This study is called Journey and is a global long-term study assessing the natural progression of LGMD for individuals with 3 sarcoglycanopathies, and most recently, we have introduced Journey for calpainopathy. We have completed enrollment for the sarcoglycanopathies, but are currently actively enrolling for calpainopathy. Journey does not involve the use of any investigational therapy, as the aim is to study the natural progression of these LGMDs and how they impact the muscles, lungs, and heart of affected individuals. In our sarcoglycan programs, we are actively enrolling into the Imagene study, which investigates SRP-9003 for LGMD type 2E. Sarepta's 9004 program for 2D is on track to initiate a phase 1 study in the latter half of 2024. And for SRP-9005 for limb-girdle type 2C, we are currently conducting additional non-clinical studies to better understand the overall safety profile before we can proceed into the clinic, which we anticipate to occur in 2025. Journey is our natural history study, and we've completed our initial enrollment in the sarcoglycanopathies, and we have now broadened the study to include individuals with calpainopathy. Before handing over to my colleague Nicole Lamarca, I would like to thank you all for your time and interest today and look forward to addressing your questions during the Q&A. Nicole, over to you.
Thank you so much, Jesse. So we just wanted to give some additional information about where you can find more information. So it's very important to know that you can email us at any time at advocacy@sarepta.com. This inbox is monitored by myself and my colleagues, and you typically will get an answer likely same day or within 24 hours during business hours. So please reach out. We also have some additional resources that have some very pertinent information for the community. So the first one here, there's a QR code for limb-girdle.com. Here you can sign up and you will receive emails allowing you to stay connected and receive information on different community resources, updated news, and information about research for limb-girdle muscular dystrophy. We also have our Genesis website. So again, another fantastic resource. There's a QR code that will link you right to the site. But if not, you can see that website right here. This gives you information on all of Sarepta's ongoing clinical trials for limb-girdle muscular dystrophy. Additionally, there's a great resource called GT Facts, and this is really looking at different frequently asked questions about gene therapy in general. So these are video series really answering questions that we've received and kind of gave answers for about the science of gene therapy, how it works, and there's just some great information there. Next slide. I think most importantly is we just want to say thank you. Thank you to the Speak Foundation for hosting us today. We were so grateful that you invited us to come speak on this wonderful study and to give an update on limb-girdle and our program in general. And we really thank all of the participants and families in our different clinical trials and for your continued interest in our work. And with that, we will open it up to questions and answers.
Great. Thanks, Nicole and Jesse. We have a lot of questions that are coming in, which is so nice and exciting. I'm going to start first with a question that's really being asked very frequently. Do I need to be retested for antibodies if I were already tested in Journey? So this is a good question. Do they need to be retested for antibodies if they were tested already in Journey?
Yeah, I'll take that one, Nicole. Yes, you will have to be tested for antibody testing. It is a requirement prior to gene therapy. So because of the timing of those tests, we have to do a test before dosing with the study drug. So regardless of whether you had a test or tests in Journey, you still have to do the antibody test again.
Great answer. Another question is about antibodies. Can I test for antibodies now in preparation to join a Sarepta gene therapy study in the future?
Unfortunately, there is no appropriate test that's available on the market beyond the clinical trials that we currently have. So outside of Sarepta studies, you cannot. So until you decide to enroll in a study, that's the only time you can test for the antibodies.
Okay. Good question here. Will tests and or procedures be covered as part of the Imagene study?
Sure, I can take that one. So all study-related costs are covered by the study. So there is no out-of-pocket cost for any study-related procedure.
Okay, good. Here we have a question. I have a variant of unknown significance, but have been phenotypically diagnosed as having LGMD. What should I do?
So I think here, and Jesse, feel free to jump in, but I think the most important thing is that you make sure that you're reaching out to your neuromuscular doctor who's very familiar with limb-girdle muscular dystrophy. It's critical to make sure that your genetic testing is recent. There are, if you are in the United States, some options for low cost testing, and you can look at those genetic testing options on limb-girdle.com. And again, it's important to have that recent genetic test. Some of those VUS that have been identified have been reclassified. So again, make sure that you're working with your doctor to see if your VUS has been reclassified. And you can go a further step and see if you can have that boost resolution. So again, working with your treating doctor, your neuromuscular doctor, is your best first step.
Right. And then here's a good question. Can I be in Imagene if I wasn't in Journey?
Short answer, yes. It's not a prerequisite to be from any of our natural history studies. So we do have within the study a lead-in period. So if you haven't, if you're not coming in with natural history data, we have a period. So the only difference here is that you will have to do some tests before you can get the study drug in the study for a 6-month period before the treatment phase can begin.
Okay, thank you. We have another question. How can I stay updated on Sarepta's work and learn about additional Imagene sites? And this is regardless of where they live, whether they live in the United States or outside of the US.
Sure. So I think a great resource, number one, is limb-girdle.com for updates. The Genesis website that we showed earlier is a fantastic place to look at kind of our ongoing clinical trials. And also clinicaltrials.gov has the most up-to-date information about where sites are, who is actively enrolling, and whatnot. So those are great options. And please feel free to email advocacy@sarepta.com and we can talk one-on-one about that as well.
Sure. We have lots of questions still coming in. We have, how much will gene therapy help important muscles like the heart and the lungs, especially for older patients?
So as we mentioned in part of our construct, we are aiming that, for affected muscles that are affected by the relevant subtype, and in this case 2E, the promoter that we use is relevant to the relevant subtype that targets those areas as well. But as part of our investigational process, we will try and understand better whether there is an improved effect on those more progressed patients.
Okay. We have a question. I think what they were asking, and I just want to try to explain it. How important is it that gene therapy for 2E expresses the full gene? Will it help, for example, muscles which are not functional?
I might need a little bit more clarification on what we mean by muscles that are not functional.
Yeah, I think that maybe this individual is perhaps thinking about like, will it help all the muscles in the body?
It's targeted for your skeletal muscles, and it can only work on parts of the muscles that are still functioning. So as I mentioned earlier, if your muscles have progressed where there is already damage, we can't undo. The aim of gene therapy is not to undo damage. It is only to stabilize and minimize the progression of what muscle you do have.
Okay. Here's a question. Do all patients have to have a muscle biopsy regardless of age?
So that is part of the clinical trial participation. Muscle biopsies are required.
Okay. Might you apply for BLA approval before the end of the 5-year duration that is based on results that are demonstrated sooner than the 5 years?
So that depends on our discussions with the regulators and what they will accept in terms of our data package. If we can go sooner with our information, and then that will be subject to the relevant regulators that we are speaking to. Different regulators have different expectations, so it's not a standardized approach that we may have to take.
Okay. We have quite a number of questions still coming through. What sorts of safety events could you anticipate that might occur? Is there anything that you can share that's been seen in similar gene therapy studies?
Nicole, do you want to take that?
Sure. So I think that it's really important to note that we are proceeding with the utmost caution when we are performing these studies. That's why, again, we're making sure to evaluate all AEs and SAEs that serious adverse events that come through. We take that very, very seriously as we participate and perform these studies. Jesse, is there anything additional you would like to add to that?
In terms of our learnings, I think that's what, as they have asked in terms of seen in other gene therapy studies, as a result of that, we take those learnings into our clinical development program. And that may include additional inclusion criteria, additional exclusion criteria, as we learn about our programs across the portfolio. So do note that that is considered as we go into the studies. And as Nicole mentioned, safety is of utmost importance. So that is a primary monitoring activity for us.
Okay. Here's a question. Is IVIG considered an active immunosuppressant treatment that would exclude participation in the study?
I would have whoever has asked that question, encourage them to have that discussion with their study physician when they consider participation in the study.
Okay. How many participants have already been enrolled, and are there open spots still?
We are currently actively enrolling in the study.
Okay, and then a patient asked how many biopsies are expected throughout the trial?
That information will be provided when you decide to participate in the study. Again, every study is different. In this study, we have a specific number. However, that may change depending on what the regulators may ask for in your specific country, as we are currently, we don't have approvals in all countries yet. So I don't want to tell you that it's a certain number. So I encourage you again to have that discussion with your treating physician when you are considering participating in the study.
Okay. When do you expect to submit the 2E gene therapy to the FDA for approval?
For that information, we are not at liberty to share that at this moment, but I encourage anyone that's interested to please follow Sarepta's information through our press releases, where you will get more information on the progress of our programs.
Okay. And then the 2E study has been clearly presented as a one-time treatment. Are you still doing any work separately on antibodies and redosing?
So at Sarepta, we are committed to trying to, again, figure out if there's an ability to explore patients who are antibody positive. So that is something that we are continuing to research as a company.
Okay. And then will there be participating centers in Europe for Imagene?
So thank you for this question. We continue to look at different site opportunities. We're working as quickly as we can with different regulatory bodies to ensure that we are moving forward with having sites activated.
Okay. Here's a question. Is there or can there be therapy for LGMD, which is not gene therapy?
Jesse, I can pass that over to you if you want.
Yeah, sure. There possibly would be, but Sarepta is not working in that space. We are currently working in gene therapy. Again, I encourage anyone that's looking for what therapies that there might be out there to look at the resources that are available, which it also includes clinicaltrials.gov, which is a more general resource that contains varied information about different studies that are occurring globally.
Okay. And then how soon will the 301 trial begin, and when will it be complete?
Well, it has already begun. So we are currently actively enrolling, and we are very pleased about that. It'll be complete once we hit the target numbers that we are aiming to obtain in order to gather the necessary data that we need to provide information to regulators. So we are targeting approximately 15 patients. So that is our target completion number.
Okay, thank you. And can the medicine be obtained under the early access program or for life-saving reasons during the phase 3 study?
Yeah, thank you for that. So at this time, there is no compassionate use, expanded access, or a right to try program for which patients can apply to. For any additional information regarding Sarepta's compassionate use policy, you can find that online. That's on Sarepta's website.
Okay. And I kind of think this question was already asked, so I'm going to skip that. What happens if one did a biopsy 10 years ago and I don't currently have a neurologist? What would you recommend for me to do in this case?
It's really important to connect with your local neuromuscular center of excellence. I think it's a great resource to reach out to the patient advocacy organization, and they may be able to help guide you to some local providers. That would be, I think, a really great first place to start. Make sure you have your genetic testing report in place, and really get connected.
Okay, that's good. Good answer. We have a question here. I'm a veteran receiving care from the VA, and they've identified dysferlinopathy as my type, and they are wanting to know what should their next steps be.
So again, I think next steps would be really connecting with a patient advocacy organization, looking at clinicaltrials.gov for additional studies that may come. You're welcome to take a look at our Genesis site and look at the clinical trial opportunities that are on that site, as well as signing up for limb-girdle.com, as there may be information forthcoming on dysferlinopathies in the future.
Okay, thank you.
Yeah.
We have a question. What are the results of patients from study 1 and 2? How are they doing now? Are patients that were non-ambulatory now walking?
We haven't yet published, and I'm assuming it means study 101 and 102. 101 was an ambulatory study and in only ambulatory patients, and we have published data on that study. We have not published data on study 102 yet. That study is still ongoing. We only completed enrollment recently for that one. And in terms of non-ambulatory now walking, I kind of talked to the results of the study. However, I want to emphasize that the aim of gene therapy is not to reverse what has already occurred. It is to stabilize and slow down disease progression. So what has already progressed in terms of your muscle functionality, we cannot confirm that whether gene therapy is going to change that. Right now, our anticipation is to slow disease progression or stabilize disease progression.
Okay, thank you. We have a patient asking that they had genetic test results from 8 years ago. They want to know, should they have it done again?
Absolutely. I think it's worthwhile to have a recent genetic diagnosis.
Okay, good answer. I am having a muscle biopsy of LGMD2E. I've not had genetic testing confirmed. So this person's saying that they've had a muscle biopsy that confirmed 2E. Should they also, could they be eligible without the genetic testing confirmation? And then could they participate is what they're asking.
So first step, they do need to have a positive genetic test. So first step would be getting that genetic testing completed.
Okay. This was kind of asked already, but I think it just is a good question. Is there ongoing science to figure out how to redose down the line?
Nicole, I think you answered that one. Do you want to go ahead?
Yeah, so I think we did talk about that. Again, Sarepta is committed to looking to see if there are opportunities and options for redosing in the future.
Okay. We have a parent says, my daughter was diagnosed with LGMD2E. She has a mutation in sarcoglycan and another mutation in YARS2 gene. Does it hinder participation in the study? Thank you. Good question.
It's a great question. And I think what I would do is first step is talk to your doctor and the study doctor for any sort of specifics on inclusion or exclusion from the clinical trial.
Okay. And is it possible that there will be early registration of 9003?
It all depends on our dialogues with the regulators. It's our intent to go as soon as we possibly can to move into that registration phase for our program. However, as I explained, there are a lot of information that we will need to provide to the regulators to assure them that we can provide a safe and efficacious product to all patients. We will progress as fast as we can, but it is subject to dialogues with the various regulators that we are working with.
Okay. And good question here. How many sites are currently open for enrollment?
Yeah, so I encourage you to look at clinicaltrials.gov for all open and active recruiting sites.
Okay. All right. And will Sarepta be expanding their pipeline to include other subtypes of LGMD?
Jesse, would you like to talk a little bit about our commitment to limb-girdle?
Well, I mean, Nicole, if you want to start, then I'll definitely add to it.
Sure. So I just want to stress there is a commitment to limb-girdle at Sarepta. We're very much so focused on our 6 subtypes, which you can see again if you look on our website. So I think that there is absolute commitment to our pipeline, which is 2A, 2B, 2C, 2D, 2E, and 2L. We have absolutely dedicated focus on these 6 subtypes in our program. We may broaden this in the future, and we will update the community if we are able to expand our portfolio. If you have, again, questions and want to stay connected, please reach out to advocacy at Sarepta, and we can talk about that.
Sure. And we have a question. Is the muscle biopsies going to be needle biopsies or open incisions? Good question.
Great question. For this study, it is all open biopsy.
Okay. What role does the Sarepta Board of Directors play, if any, in deciding when to submit a research packet to regulators?
Sorry, can you give me a little bit more clarification to that question?
Yeah, I think they're asking, do the Board of Directors have part of any decision-making when it is sent over to regulators for approval? I think that's kind of the question. Do they have anything to do with that, or is it more based just in your scientific area, I think?
Well, it's based on the scientific rigor of what we can provide to regulators. We obviously have a governance process within each organization that assures that we are taking the most appropriate programs through the pipeline. However, it is the data that drives our conversations with the regulators, and it is their assessment of that data that gives us guidance as to whether our product is approvable or not.
Okay. Okay, and we have this kind of question's good, I think. Can you confirm that the Sarepta investigational treatment can only pause the muscular decline as opposed to rebuilding muscle? So I think this question is, can the gene therapy treatment rebuild muscle, or does it just stop progression? I think that's the question they were asking.
So, Nicole, I'm gonna start, but please feel free.
Okay, sounds good.
Our aim for gene therapy is to slow or stabilize disease progression. Pause, I guess, is the word that was used. Rebuilding muscle is ideal, but we are not able to confirm whether that's what our gene therapy will or will not be able to do.
Okay.
I think you covered it well. Thank you, Jesse.
Yeah. Is it plausible to hope that gene therapy for 2E will be commercially available by 2026 if trials go well?
I love the angle of question, I guess. It really does depend on how much, how fast the studies can enroll, how fast we can get the data, how fast we can dialogue with the regulators, and whether we can be successful with that data package to the regulators. As soon as we have information, the community will know, through the community letters that we send out, as well as the press releases that Sarepta provides to the wider community as to the progress. So, as soon as we are able to share that information on those timelines, we will.
Okay, thank you. Boy, you guys are getting a lot of questions, aren't you? All right. Do you have to be 18 years old to participate in the trial? That's a question on the ages, which is a good question, I think.
No, the minimum age to participate in the study is 4 years and older. So in terms of our inclusion, you have to be a minimum of 4 years. So this is not restricted to adults only.
Okay. We have a question here. I'm a 58-year-old adult in the UK with LGMD 2A. Is there an eligibility requirement regarding the age for the current calpainopathy study that was mentioned? So I believe they're asking that question. Good question, asking another age-related question there.
This is for the Journey study. For the Journey study, we have no age restriction for participation in the study.
Okay.
I will just add to that. I encourage you to look at clinicaltrials.gov for that, also the Genesis website, and connect with additional advocacy organizations such as Speak Foundation.
Yes, absolutely. And calpainopathy, also there's C3 as well that could help with that. And then,
thank you, Kat, for mentioning that.
Sure. And then can you please explain difference between Journey and Emergence? And I think when they say Emergence, I think they mean Imagene.
Sure. Journey is our natural history study. So this study is just studying the natural progression of your relevant limb-girdle subtype. And in the case of Journey, we were investigating 2D, 2C, 2E, and now most recently 2A. So that is a non-treatment study, and it just follows the patients on how their disease is progressing. Imagene is our phase 3 study for SRP-9003, which is our product for LGMD type 2E or R4 for beta-sarcoglycanopathy. So that is a treatment study, an open label study, where we are going to investigate the safety and efficacy of SRP-9003.
Okay, thank you. Thank you, Jessie. Well, I think we're at the end of our time on terms of questions. I see that we may have more questions. One of the things we want to make sure is specific questions about your medical diagnosis are things that we can't necessarily answer on air. So just please consider that there's some questions that we weren't able to answer on air because they were specific to HIPAA, and we have to be cautious about that. So I want to say thank you to Sarepta and their team for being with us today. It's very exciting, and we appreciate your time. And again, if you have any questions, you can refer them over to advocacy at Sarepta. And if you have any other further questions on how to connect with them, feel free to reach out to Speak Foundation. And we will hope to see you on our next webinar. Thank you so much for being with us, everyone, today.